Okay, hello everybody. So this is the Genetics of Complex Disease lecture from the Human Genetics module, which is a level 1 module on the Biomedical Science degree course at the University of Bradford in the UK. So the idea of this lecture is that by the end of the lecture you'll be able to discuss the genetic component of complex disease, explain conceptually how we understand how many genes can influence a complex disease trait and the likelihood of actually developing a complex disease, and then talk a little bit about how we're able to experimentally determine which parts of the genome might be responsible for this genetic component of these complex diseases using specific examples from the lecture. So we'll start by discussing what exactly a complex disease is, what I mean by complex disease. I'll give you some examples of those, and then ask the question, why are we interested in trying to investigate the genetic component of these particular diseases? What do we hope to benefit and gain from understanding more about the genetics of these complex disease traits? How do you actually know in the first place if a complex disease has a genetic component? How can we tell if genetics is involved at all or whether it's solely environmental? We'll talk a little bit about polygenes, what they are and how they contribute to complex genetic disorders, before going on to some of the analyses which have been used historically and are being used currently to try and identify players that are involved in the susceptibility of ourselves to these complex disease traits. And these include linkage analysis, and we'll go on to discuss the HapMatch project and so-called Genomewide Association studies, which are the sorts of studies which are ongoing at the moment in the literature. And we'll finish with a summary of the lecture. Okay, so what is a complex disease trait? Well, this is a little bit of an obvious statement. Complex diseases are diseases that are not monogenic, so a little bit of a nothing statement. Let me expand on that somewhat. So what I'm talking about is diseases which are not caused by mutation to a single gene. So we've talked a lot in the last lecture about mutations in single genes which give rise to disease, such as cystic fibrosis, Huntington's disease, achondroplasia, sickle cell anemia. All of these diseases are really horrific diseases in a lot of cases that arise due to a mutation event in the gene of a single allele. Yeah, so a single gene. Complex genetic diseases do not arise in the vast majority of cases due to a mutation in the single gene, although there are one or two exceptions which we'll talk about in this lecture later on. But the vast majority of these diseases do not arise because of a mutation in a single gene. So complex diseases are the big diseases, if you like, so the big players in world health, global health, such as cancer, asthma, migraine, insulin-dependent and insulin-independent diabetes, arthritis, multiple sclerosis, hypertension, heart disease, obesity, Crohn's disease, and then also some of the, the neurological disorders and social disorders, learning disabled disorders, etc., such as autism and schizophrenia, and also neurodegenerative disorders such as Alzheimer's. So these are the big players. These are what are going to really finish most of us off. I think at latest count, 40-50% of the people in this lecture theatre will probably die of a heart attack or heart disease. Another 35% or so, a third of you will die of cancer. These are the diseases which are going to end up causing us our death, and obviously therefore they're of incredible interest with regards to medical research and the healthcare profession. So the other reason they're of interest is, one, they're far more common than single gene disorders. We've discussed at length how rare single gene disorders are, how some of them actually only affect a handful of people across a globe, across a planet. Very different with regards to complex disease. These are the diseases which affect everybody in every country to a very high extent. And also because of that, they incur significant healthcare costs. It's thought that complex genetic diseases account for around £100 billion a year in the UK alone with regards to healthcare to manage and try and treat these diseases. So a couple of years ago there was a lot of work and a lot of media coverage about the cost of caring with people with dementia, and the fact that around 1% of the gross domestic product of the country was used, of the world, was used in dealing with and treating and providing care for patients with dementia, which is another form of, often caused by one of these complex disease traits. So there's a huge financial burden on the world as well with regards to maintaining and treatment of and caring for people who suffer from these disorders. So where does genetics fit into this? Why is it of use to actually try and understand the genetics of these complex diseases? As we're going to discuss, it's very difficult to actually pin down how individual genes are contributing to or leading to an increased susceptibility to these diseases. It's very rare that there's a simple answer that's out there that can directly lead to a greater understanding or a cure, if you like, for these illnesses. So why bother studying the genetics at all? Why not just try and treat the illness? Well, there are several good reasons as to why we need to try and understand the genetics in more detail. The first reason is so that we're able to identify those people who are at increased risk for actually developing these diseases later in life. So if we're able to identify increased susceptibility in patients, then that might allow that patient to impose a lifestyle change that could have a drastic effect on whether or not they end up actually developing that disease or not. So if we tell somebody, for example, look, you've got a very high susceptibility to heart disease, that person might decide not to go to Subway so much, but to opt for a salad instead, or do more exercise. And as we've seen with other genetic diseases, changing the environmental factor can have a huge effect on whether or not the disease manifests or not. And that's particularly true for these complex diseases where the environment plays a large part in the susceptibility of and the actual establishment of disease in patients. The second reason to study them is that it's not always clear from a set of symptoms or from a disease what the underlying molecular causes are that have caused that disease to arise. So that is to say that diseases which look very similar with regards to symptoms can have very different etiological pathways. So etiological is a posh word for cause, so it can arise due to very different causes. So we now understand this is true for all sorts of different complex disease. For example, skin cancer, melanoma. Melanoma looks very similar, but we now understand that actually malignant melanoma is probably caused via lots of different molecular events, and that these different molecular events actually require a very different approach with regards to treatment. So this is an explanation as well for medics and for obviously clinical scientists. This is of interest to you. This is one of the explanations for why certain patients respond well to one regime of treatment, but not another set of patients will not respond so well to that regime. And a better understanding of the molecular pathways which lead to complex disease will enable clinicians to actually prescribe drugs which are going to have a good effect for that particular type of disease in that patient. So it's towards if you like, a more personalised pharmacogenomics for the individual, where we understand more about the molecular and genetic basis of a complex disease, and from that understanding can have a more directed therapeutic approach with regards to what medicines to give that individual. Finally, understanding more about the genetics of a disease might give us information about particular genes or proteins which are involved in the disease, which are involved in causing the disease, which previous to this we didn't understand had a role in that particular illness. And the identification of novel proteins which are involved in causing the disease may offer the opportunity to develop novel drugs which can treat diseases in a new way and perhaps a more effective way than the current drugs are able to do that are used at present. So there's three good reasons there for really trying to study complex disease and the genetics of complex disease with quite a lot of urgency. So when we study genetic disease and complex genetic disease, what are we looking for? Well, one of the main things you need to get your head around when we're talking about genetics and complex disease is it's not like single gene disorders where we're talking about a mutation in a gene causing the disease. So in cystic fibrosis, a mutation in the cystic fibrosis gene has an effect on the cystic fibrosis protein that causes a cystic fibrosis disease. There's a direct relationship there. When we talk about genetics and complex disease, we're often talking about risk, increased risk of, increased likelihood to go on and develop. And it's these risk alleles that we're actually searching for in populations and in families to try and identify aspects of our genome, aspects of our DNA sequence that appear to give an increased likelihood that we're going to go on and get cancer, that we're going to go on and get heart disease, etc. And traditionally, these risk alleles have been identified via two different methods: parametric and non-parametric linkage studies. And we'll talk about what those are later on. More recently, after the advent of the Human Genome Project, and with the availability of increased sequencing technology at lower prices and more reproducibility, we're now able to do much more powerful population-based studies using a process called Genomewide Association studies, which we'll talk about again towards the end of the lecture. So how do we know then when a complex disease has a genetic component? Well, here's another definition for you. So the proportion of variation in a trait that is due to the genetic variation is called heritability. And heritability is generally defined as a number between 0 and 1. And as this number increases, so does the amount that the actual trait, the phenotype, is influenced by the genetics. So for example, if you had a trait which was completely environmental in nature, its heritability would be 0. If you had another trait that was completely genetic in nature, and no matter what happened in the environment, it was the genes that determined this phenotype, that would have a heritability value of 1. Now, it's a little more complex than that because it's not always the case that a high heritability factor means that the phenotype is solely determined by genetics. And the classic example of this is height in humans. So obviously some humans are very tall and some humans are very short. And there's been a lot of studies to show that heritability of height is actually quite high, about 0.8. So in other words, the height that we are is largely determined by our genes. But it's not as straightforward as that, because if you look at average height across populations over the last 100 years, what you see is that average height has actually increased quite a lot. Increased far much more than we could attribute to evolution via genetics. So what that tells us is that whilst there's a strong heritability factor for height, in other words, your height is largely determined by your genes, the ability to actually get taller can also be influenced greatly by the environment, because it's environmental factors obviously which have caused globally across many populations height to increase, improved nutrition, etc., improved healthcare. So this is an important ramification for complex disease, because it tells you that even complex diseases which are known to have a very strong or very high heritability factor can still be really drastically affected by uniformal changes in the environment. So that's to say that, for example, if somebody told you, oh, I'm really sorry, there's a very, very high likelihood that you're going to go on to develop heart disease because it runs in your family and there's a high heritability factor associated with that particular complex disease, that doesn't mean that by changing your environment, by doing more exercise, by eating less fat, you can have a drastic effect on that actual trait, aside from the fact that there's a strong heritability factor there. So how do we know then when a complex disease has a genetic component? Well, historically, these kind of questions were answered by studies called twin studies or concordance studies. And what these studies did is they took advantage of either monozygotic and dizygotic twins with regards to working out whether or not there's a genetic component to particular diseases. And the idea is that dizygotic twins, non-identical twins, share 50% similar genetic sequence. So their genomes are 50% similar. Same as you and a fraternal sister is, or a paternal sister. Monozygotic twins, in theory, are 100% genetically identical. So these are two individuals who've come from the same egg, same set of chromosomes. Yeah, so they're genetically identical. So if we compare the prevalence of certain complex diseases between dizygotic twins and monozygotic twins, if there is an increase in the actual prevalence of that disorder between dizygotic and monozygotic twins, that would suggest that there's a genetic component contributing to that actual disease. So this is shored up by the idea that these twins as well would have been raised by the same family in the same environment, so being born from the same womb. So largely the environmental factors are pretty static for these people. So any difference is observed will be due to genetics. Now that's a very naive view, because very subtle differences in the environment can have very large effects, obviously. But let's run with it for the time being and have a look at what the data actually shows. So if we start with something like breast cancer, what we see is that the prevalence of breast cancer between dizygotic twins and monozygotic twins is quite similar. There's no increase there. So if you're a monozygotic twin and your sibling develops breast cancer, there's very little evidence to suggest that you're also going to go on and develop breast cancer as well. Yeah? So quite a weak genetic component to breast cancer. However, if we then look at type 1 and type 2 diabetes, what we can see here is that dizygotic twins, very little concordance between the likelihood of developing these diabetes if your sibling develops it. If you're genetically identical to your sibling, however, in particular for type 2 diabetes, if your sibling develops type 2 diabetes, you develop type 2 diabetes, suggesting that this extra 50% genetic similarity is what's actually accounting for this, very strong concordance for this particular disease. And indeed we see strong concordance figures for other diseases such as arthritis, which is a complex disease, and also tuberculosis. So if you're a monozygotic twin, the likelihood of you developing the disease, if your twin does, is increased for arthritis and tuberculosis, suggesting there's a strong genetic component contributing to the likelihood of getting those diseases. Now, stop and think about this table for a second. For the case of tuberculosis, this is particularly interesting because the other diseases here are autoimmune diseases largely and then cancer. This isn't actually that type of disease. Tuberculosis is caused by a bacterial infection. So surely bacterial infections should be completely environmental. The disease is caused by something from the environment. So why is there such a strong genetic component? Well, this relates to the fact that the actual resistance, our innate resistance to pathogens, is itself often controlled by numerous genetic loci, by our genetic makeup. And that historically it was infection by pathogens that was a big killer, a major source of morbidity in across populations. So it's not, ridiculous to imagine how through evolution certain genetic traits have been selected for which provide an increased resistance to infection with certain pathogens, and that if your sibling has developed tuberculosis because they lack these protective genetic loci, then if you're an identical twin, you're also going to lack those protective genetic loci, and therefore your susceptibility to TB will also be increased. So it's not just like physical diseases, if you like, that have been analysed using these concordance studies. We've also used them to analyse psychiatric and behavioural disorders. So one of the real powerful results that came out of this work was the evidence that if you look at concordance studies for autism, for example, there's a really strong genetic component present for autism in children. Now, this is important because historically autism was actually blamed on poor parenting or loose parenting, middle class parenting, if you like. And that caused a lot of trauma and upset, obviously, to the parents of autistic children who were blaming themselves, thinking that it was their fault, they'd done something wrong. But actually, we understand now that autism is probably resulting due to a strong genetic component. So it's something that the parents have passed on in their genes to their offspring, which obviously they can't help. It's not something they've done with regards to the parenting skills. There's strong genetic evidence, a strong genetic component for other diseases such as Alzheimer's and schizophrenia in particular. Less so for alcoholism and dyslexia, but it's still present. It still seems that there are genes which are contributing to these disorders as well. Now you can take this to the nth degree, and in fact there have been some controversial studies where the people have tried to use concordance experiments to demonstrate a genetic contribution to just more general traits, violence, breaking the law, aggression, things like that. And those kind of studies are, quite controversial, and a lot of the data is often misinterpreted. But the stuff I'm showing you here today is pretty much accepted now in the literature that things like autism and schizophrenia have a very strong genetic component to them. So these are complex diseases which have an underlying genetic element to them. So another way of classically studying then how genetics are contributing to a disease are via these family studies or familial studies. So these are slightly different to concordance studies with identical twins, but share some conceptual similarities. So if you think about degrees of relatives in your family, so let's start with first degree relatives, which are either your mum and dad or your children or your brothers and sisters. So your mum and dad, your children or your brothers and sisters are first degree relatives, and in effect those individuals share 50% of the alleles that you have. Yeah? So you are 50% from your mum, 50% from your dad. Your children are 50% from you, 50% from your partner. And likewise, you are going to share around 50% of the alleles that your brother will have or your sister will have. Similarly, second degree relatives like grandparents, aunts, uncles, etc., they're 25% similar to you. And third degree relatives like cousins, great grandparents, etc., 12.5% relative to you. So, what scientists have done, what people have done who are interested in trying to determine whether complex diseases have a genetic component, is they've looked at these diseases and seen whether or not there is an increased likelihood of inheriting disease in the offspring of individuals with this disease, and whether that increased likelihood correlates to the relatedness of that individual to the person with the original disorder. Now, adoption studies are particularly useful in regards to this, because what you're able to do with the adoption studies, at least in theory, is dissect nature from nurture. So this includes a classic example whereby children who had been born to schizophrenic mothers were adopted by another mother for obvious reasons. Perhaps the original mother was, the biological mother was, just not well enough to look after the child, so the child was put up for adoption. But if there's a genetic component to schizophrenia, then a child put up for adoption who's been born from a schizophrenic mother would have an increased likelihood of themselves being schizophrenic. If there's no genetic component to schizophrenia, an adopted child from a schizophrenic mother should be no more likely to develop schizophrenia than any other child in the population. So what do you actually see? Well, if we look at the actual likelihood of developing schizophrenia, let's have a look at this. So if you're just an unrelated person in the general population, so the majority of people who are listening to this in this lecture theatre, there's a 1% chance that you may develop schizophrenia. Now, conversely, if you're an identical twin of somebody who has schizophrenia, who's been diagnosed with the disorder, there's a 50% likelihood almost that you're also going to be diagnosed with schizophrenia during your lifetime. So that's very strong evidence that there's a strong genetic component to this disorder. Similarly, if you're the offspring of two schizophrenic parents, other than having a very interesting childhood, I would imagine, you're also very likely, 50% likely, to go on to be diagnosed as schizophrenic. And you can see that this likelihood decreases if you go to second degree relatives, if you like, so reduced genetic similarity, whereby siblings, fraternal twins, offsprings of one schizophrenic parent, there is a reduced but still elevated risk of getting schizophrenia above and beyond the general population. So these family studies can be used to show where the likelihood of developing a disease is higher than would be expected in the general population if that disease runs in your family. So we've talked a lot about these risk factors, genetic factors, etc. What exactly are they? How do they fit into our model of genetic inheritance, for example? And in fact, the concept of the genetics of complex traits caused a lot of friction between two of the heavyweights of genetics at the turn of the 20th century. Francis Galton, who, if you recall from the introduction lecture, was the eugenics guy who promoted eugenics, Charles Darwin's cousin, very intelligent man, debatable ethics. And William Bateson, who was the guy who liked to surround himself by female scientists at Cambridge, and who also kind of rediscovered Mendel's work and brought it to the masses. So we had Francis Galton, who really was interested in the genetics of complex traits, if you like, general traits in the population through his eugenics associations. So he was interested in the genetics of aggression, the genetics of behaviour and things like that. And William Bateson was a classical geneticist who believed that Mendelian genetics just was the only type of genetics that was of worth, and that everything must obey Mendel's laws of inheritance, independent assortment, etc. So these disputes, these arguments between Bateson and Galton were eventually settled in 1918 following publication of some work by a guy called Ronald Fisher. And what Ronald Fisher described for the first time was a concept of polygenes. So what Fisher said was, if you consider the possibilities that arise from a particular gene, so let's start with one gene here, and if you think about the different ways in which the alleles can be assorted, in this gene, there are three really that you can get, three situations. You can get, if you like, big A, little a, and little a, little a. And if you remember from your Mendel's first law, the ratio of this is 3:1, or you get you get one big A, two big A, little a's, and then one little a. And so effectively you form this kind of distribution. Now, that's for one gene. If we increase the number of genes to 2 genes, and look at the different possible genotypes, you generate this kind of pattern. So now this is the frequency of the different possible genotypes. If we go to 3 genes, this is the frequency of the different possible genotypes. And you can see now where we're heading. We're heading towards a situation where if the number of alleles that are actually impacting on a trait become large, so for example if we've got 100 alleles here instead of just 3, the curve that you actually end up producing is a curve of normal distribution. So this is a way in which we can still use Mendel's laws of segregation and inheritance that Bateson championed, but we can use them in such a way that we can explain the sort of distribution of a trait that we would see for something like height or weight across a general population. So Fisher's work allowed Mendel's laws to stay intact, but also allowed the possibility that genes were contributing to the distribution of if you like, a complex trait such as height. Excuse me. So a problem with this obviously is that if you think about most diseases have a discontinuous distribution. So you don't just have a little bit of cancer, you either have cancer or you don't have cancer. So how does this concept fit into the polygene situation then? Well, how this fits in really is via something called the liability threshold. So what the liability threshold states is that when alleles push the distribution of a curve towards the liability threshold, they are termed risk alleles or susceptibility alleles. And that when you go past this liability threshold, that's when you actually manifest a disease. So if we think of something like heart disease, this is a normal distribution curve for heart disease, which represents all the alleles and the likelihood of that contribute to this disorder, and also the likelihood of actually having heart disease in the population. What we see is only a small proportion of the population actually have the disease itself, and this proportion of the population are represented by individuals who happen to have a large number of the susceptibility alleles required for that particular disease to manifest. Now, if this model is correct, if individuals with lots of susceptibility alleles end up manifesting the disease, then it stands to reason that the offspring of affected individuals would themselves be more likely to be affected because they would be inheriting lots of susceptibility alleles. Yeah. Now we can actually prove that this model is correct, the liability threshold, or it suggests strongly that it's correct, via looking at some of the data around a particular congenital disorder called pyloric stenosis. What pyloric stenosis is, it affects newborn babies and causes them to vomit violently due to some issue with the digestive tract. But the interesting thing about the disorder is that it's 5 times more likely to occur in boys than it is in girls. So what you can infer from this is that the liability threshold then for this particular disease, for pyloric stenosis, the liability threshold for actually getting this disease is higher in girls than it is in lads, or boys, sorry. So in other words, if you're a boy, you may only need to get to this point on the curve, and then you'll develop the disease. If you're a girl, you only develop the disease if you get right to the end of the curve, if you've got a lot of these susceptibility alleles present. So if we take that a little further, if you think about an affected girl, so a girl who has suffered from pyloric stenosis, who then goes on to have a child, or have children. If she's been affected, this woman, by pyloric stenosis as a baby, it suggests that she's operating at the very end of this curve. In other words, she must really have a lot of these susceptibility and risk alleles present in her genome in order to have manifested that disease. And therefore, when she has a child and passes on that large proportion of susceptibility alleles, her offspring, her children, should be at a much higher risk of actually getting pyloric stenosis than the rest of the general population, or from the offspring of a mother that didn't suffer from pyloric stenosis. Yeah? And that's exactly what we see. We see that mothers who suffer from pyloric stenosis themselves, who then go on to have children, their offspring are 5 times more likely to actually suffer from the disorder than the offspring of a mother who didn't have pyloric stenosis. So this confirms that complex disease traits often arise due to the accumulation or the presence of many of these susceptibility or risk alleles being present in the individual. Now it's a little bit more complex than I've outlined here. Not all alleles just work in a simple additive manner. Some alleles are dominant over other alleles. Some alleles display epistasis. In other words, the presence of one allele can affect how other alleles may operate in the system. Some are protective. So you've got a very complex network of interactions going on between lots of different points within the genome, all of which contribute towards either pushing a phenotype, a trait, towards a disease state, or protecting an individual from that threshold point and the subsequent disease that's associated with going past it. So I hope that's clear. There's a good chapter in this on one of the recommended textbooks, Human Molecular Genetics by Sudbury. In fact, a lot of this lecture is based on work from that book, so you might want to look at that in the library. Okay, so how do we actually find these disease alleles experimentally then? Well, historically, single gene disorders were identified using a technique called parametric analysis, parametric linkage analysis. It's called parametric because you have to specify the parameters of the disease in order to be able to use this type of analysis. And what you're effectively looking for is co-segregation of a particular part of the genome with the disease state. Because you need to know the genetics, however, of the disease in order to be able to actually carry out this type of analysis, it's pretty much useless for trying to determine parts of the genome, gene loci, which are important in susceptibility for complex disease. Because the point of complex disease is we don't understand the genetics of it. We don't understand how all these different susceptibility factors and genes are interacting to push a disease towards a liability threshold or protect it from the liability threshold. However, there have been some examples where this technique's been used and identified key important genes which can give rise in rare occurrences to complex diseases. So for example, there are cases of where complex diseases such as breast cancer, Alzheimer's disease and heart disease have all been analysed using parametric analysis and identified specific genes which are involved in the causative effect of those particular diseases. Now, importantly, these only work when the complex disease is actually being caused by a single gene defect. So it's a bit of a cheat, really. These are diseases where the complex disease isn't actually caused at all by the predisposition and the combination of environmental and genetic factors. It's almost like a complex disease that's caused by a single gene mechanism. So in the case of breast cancer, the parametric linkage analysis identified a particular gene called BRCA1, and there's also a second gene called BRCA2, which seem to segregate with cases of breast cancer in families. Now, importantly, this was breast cancer in families which demonstrated an autosomal dominant mode of inheritance. So the mode of inheritance immediately suggested a Mendelian single gene effect, and that's what was going on. There are similar cases for Alzheimer's, where very early onset Alzheimer's, which is unusual, so this is where people who are quite young get the disease, have a mutation in the ApoE gene and other associated genes, which have subsequently been shown to be able to form a very aggressive type of Alzheimer's disease. And similarly, mutations in one of the low-density lipoprotein receptors are associated with premature heart disease. So in each case, it's a complex disease that's been caused by a single gene defect. And these players, if you like, these genes which are involved in the manifestation of these diseases were identified using parametric linkage analysis. So there's a second type of parametric analysis called non-parametric analysis, LOD analysis, called non-parametric analysis. And this is a type of linkage analysis that analyzes co-inheritance of regions of the genome. And the basic premise is that if there's an allele or a region of the genome that's contributing towards the likelihood of a particular complex disease, then the actual part of the genome within which the affected gene is situated will be co-inherited from an ancestor in a family tree who was affected by that disease by other affected members within the same family more frequently than would be expected by chance. So that's a little bit of a complex way of putting it, but I'm not sure how to put it any clearer than that. It's called non-parametric analysis because to make that statement and that conclusion, you don't need to know anything about the actual genetics or any parameters to be actually known beforehand, like you do with parametric analysis. All you're assuming is that if there's a region within the genome within which the affected gene is contained, then that particular region of the genome will be inherited from if you like, affected ancestors by affected offspring at a rate higher than what would be expected by chance, i.e. 50%. So initially these regions were tracked via PCR analysis of microsatellite regions of the genome that basically allowed scientists to chop the genome up into about 300 bits, look at each of those bits and ask whether a specific piece of the genome that was present in an affected individual was always found at an increased rate in affected offspring. And if it was, then that particular region of the genome was thought to perhaps be involved in the actual etiology, the actual cause of the complex trait being studied. So this type of analysis was used by a guy called Davies in the early 90s to describe a real strong association with the HLA locus and an increased risk of type 1 diabetes. So what Davies saw was that the HLA locus, alleles for the HLA locus, were really strongly inherited within families and also within populations between affected individuals, suggesting that the HLA locus was somehow important for, predisposition to diabetes, to this particular disease. Now, the HLA locus is the part of the genome that's responsible for presenting different antigens on the cell surface, so it's not surprising that it might be important in the manifestation of an autoimmune disease. And it shows you how non-parametric linkage analysis combined with population studies it's quite a powerful way in some ways of identifying these risk alleles, but it's also severely limited, and its main limit is numbers. Even with population-based studies, it's difficult to look at enough individuals within a population and certainly within a family to identify alleles which may only confer a very small increased risk to developing the disorder. So the HLA locus has a very big effect on the likelihood of developing diabetes, but there are probably many other alleles which confer a very small risk to developing diabetes, and in some ways it's these many dozens or hundreds of alleles or thousands of alleles or whatever it turns out to be, this very small contribution that when added up represents the bulk of the risk associated factors that drive people towards this liability threshold. So what we really need is a way to identify these very subtle effects that our genome is having on our likelihood of developing complex disease. So we're now getting closer to being able to actually look at these risk alleles in that level of detail via work that was born out of the Human Genome Project. So one of the things that the Human Genome Project revealed was the presence of these so-called single nucleotide polymorphisms, or SNPs, as they're abbreviated to. And what these single nucleotide polymorphisms are differences in individual nucleotides between individuals. So these are single nucleotide changes all across the genome that vary between person to person. So one person, for example, may have a C at this particular chromosome. Another person may actually have a G at this particular chromosome. Another person may have an A at this particular chromosome. Each of these SNPs, these single nucleotide polymorphisms, differs throughout the populations, and they differ at a relatively high percentage. So what a project called the ENCODE project looked at were SNPs that were more than 5% prevalent in the general population. So for example, if 95% of the population had a C at this residue and 5% of the population had a G, then that G single nucleotide polymorphism will be included in this ENCODE analysis of the human genome sequence. And what this analysis revealed was that there's an unbelievable amount of these SNPs present in the human genome. 11 million, between 11 and 15 million single nucleotide polymorphisms which are present within a genome, and which each single nucleotide polymorphism is potentially different from person to person in different combinations. So if there's 11 million points within the genome that can be different, you can start to see already the dizzying amount of potential variation that already exists within our genome with regards to these single nucleotide polymorphisms. And the idea is that these differences in single nucleotide polymorphisms might explain some of the differences which we observe with regards to susceptibility to disease. And that if we can identify certain patterns of these single nucleotide polymorphisms which are associated with an increased risk of developing that disease, then we can track and predict susceptibility in other people by sequencing these single nucleotide polymorphisms within the genome. Now it goes a little bit further than that, because following on from this work by ENCODE, there was a project carried out called the HapMap project. And what this project realised was something really interesting. Now obviously if you're trying to analyse all 11 million single nucleotide polymorphisms within someone's genome, you've got a huge task there to track that amount of information. Simply not feasible. Even now with modern next generation sequencing, that would be an incredibly labour intensive thing to have to do, and certainly almost impossible to interpret. But it turns out that if you actually look at differences in the single nucleotide polymorphisms that appear across our genome, one or two single nucleotide polymorphism changes can actually inform you as to what a lot of other surrounding single nucleotide polymorphisms will actually be for that person. So in other words, if you look at a block of SNPs and identify the nature of one or two of those SNPs within that block, depending on what the result of that experiment is, depending on what those one or two SNPs look like, if it's an A and a T or an A and a C, that then allows you to infer and predict what a lot of the surrounding SNPs will be. So it seems that these SNPs are kind of inherited as so-called blocks, or haplotype blocks within genomes. So rather than having to sequence all 11 million SNPs to work out what someone's SNP profile would be, you can actually get away with sequencing just a few thousand, and from that data infer and make a good guess at what the remaining SNPs will be for that individual. So this then allows the generation of these so-called HapMaps, these haplotype profiles for individuals within populations, and the start of something called Genomewide Association studies, which are becoming ever more prevalent due to continuing advances in DNA sequencing technology. So what these Genomewide Association studies do is they look at a particular pattern of these SNPs and see whether a particular pattern of SNPs is more prevalent amongst affected individuals within a population than another pattern. So if there's a certain combination of SNPs, for example, that always seems to to be present with people who develop heart disease, then it could be the case that the genes that were contained around and within these SNP regions are important for the manifestation of that complex disease. And the fact that these individuals have inherited this particular pattern of SNPs corresponds to the inheritance of a particular genetic region, a particular gene that might be involved in the etiology of that complex disease. Okay, so there's lots of information about genome-wide association studies in the media. We're forever hearing news reports about scientists have identified a new gene that's involved in obesity. Scientists have identified a new gene involved in Alzheimer's. Scientists have identified a new gene involved in ADHD, as we see here. There's a genetic link, blah, blah, blah. And a lot of these reports you see in the media are born out of these genome-wide association studies. The fact that people have looked at a haplotype for a large number of people, seen that a certain combination of SNPs are always found in these individuals with this disease. We've then looked at what region of the genome those SNPs correspond to and seen that a particular gene is found within that region. And then we've said, well, this gene must be involved in the actual development of this disease. And then the news people get hold of it and things get, broadcast to the masses. The reality is you always need to be very careful about inferring function to genes identified via genome-wide association studies, because what you're dealing with are genes which may have a very small effect on the actual disease itself. So whilst there might be a correlation there, we don't know how strong that correlation is. Certainly it's unlikely to be as strong as a correlation that was identified for HLA using non-parametric analysis. It's likely that it's just one of many, many different factors that are involved in development of that particular illness. So it's very important that you always keep a close eye on whether or not something's genuine or not. We know for a fact that what's apparently the truth doesn't always turn out to be quite so clear cut. So just to summarise then, complex diseases are the big players in healthcare: cancer, heart disease, diabetes, etc. And we need to understand the genetics of these complex diseases because we can improve diagnosis and treatment of the disease if we can diagnose it early in people by spotting susceptibility. If we can understand how these complex diseases are working at the molecular level, perhaps we can give different treatments which are appropriate for different types of disease. And also identifying genes which are involved in these disorders may lead to the development of future therapeutic regimes. Historically, people have identified a genetic component of complex disease via twin studies, concordance studies, and how exactly you can marry Mendelian genetics with complex traits and continuous variation in populations, well, that's the work of Fisher, his work on polygenes, and the idea of this liability threshold that exists for whether or not people actually manifest the disease or not. Linkage analysis pioneered the discovery of a lot of susceptibility genes, but there were issues with it, statistical issues, and it's now been replaced by a more powerful techniques that allow us to look at variation across the entire human genome in the form of these SNPs, these haplotype blocks, and this has led to the formation of many genome-wide association studies which are capable of identifying parts of the genome which always seem to be present when a person develops a certain disease. And this is an ongoing area at the minute to help try and identify other players which are leading to heart disease, cancer, and other big killers and big diseases which impact on social health. So I hope this has proved useful. I appreciate that aspects of this are difficult to follow in a podcast, particularly the parametric, non-parametric linkage analysis. I would urge you to look at the core textbooks for those sections and read them. Have a look at the figures and see if you can understand that in a little bit more detail than perhaps I've been able to go into in this lecture. Okay, thank you very much, and look out for more lectures in the series.